Hi, we are member from Group 3, Subgroup 4. So my name is Tan Sing Ro, matric number 1063350.
My name is Yosini, matrix number 106745.
My name is Aisha Danira, matrix number 97234.
My name is Alicia Gabriel, matrix number 103230.
So in this video, we are going to present practical 2A and 2B, which is spectrophotometry reducing sugar analysis using DNS method and determination of sugar in sample using DNS method.
This experiment objective is to prepare a glucose standard curve using a spectrophotometer and to quantify the sugar's concentration in the given samples using the DNS methods. In this practical session, the materials used is the distilled water, DNS reagent, Rochelle salt solution, standard glucose solution, 10 gram per liter, and also the sample solution. And for the apparatus, is the cuvette, glass beaker, hot plate, laboratory mixer, micropipettes and also the tips, spectrophotometer, and the test tubes and also the test tube racks.
For practical 2A, 6 clean and dry test tubes were prepared and labeled. The glucose solution, distilled water, and DNS reagent were added into the test tubes following the table. Distilled water addition, DNS reagent addition, glucose solution addition. The mixture is then shook well to ensure the complete mixing. The test tubes were boiled for 5 to 7 minutes. Then, the test tubes were cooled down to room temperature. After cooling, 1 ml of Rochelle salt solution was added in the mixture. 2 ml of each of the mixture were transferred in a cuvette. Each cuvette were then placed in the spectrophotometer at 575 nanometer absorbance. The absorbance readings were recorded, and the graph of the absorbance against glucose concentration were also plotted. This practical was done in duplicate. For practical 2B, 6 clean and dry test tubes were prepared and labeled. The sample solution was diluted with the ratio of 1 to 9, which is 1 ml of the sample solution and also 9 ml of the distilled water. The sample solution, distilled water, and DNS reagent were added in each of the test tubes following the table. Sample solution and distilled water addition, DNS reagent addition. The test tube is then boiled for 5 to 7 minutes. The test tubes were cooled down to room temperature before adding the Rochelle salt solution. Then, 1 ml of Rochelle salt solution was added in the mixture. Rochelle salt solution functions in blocks the dissolved oxygen from oxidizing to its original yellow color. This is important for the absorbance reading later. Then, 2 ml of each of the mixture was transferred in the cuvette. Each of the cuvette were placed in the spectrophotometer at 575 nanometer absorbance. The test tube 6 will act as a blank to eliminate the background readings. This practical was done in duplicate.
In conclusion, this experiment successfully constructed a glucose standard curve using the DNS method and UVV spectrophotometry. The results show that the absorbance increased with glucose concentration at 575 nanometer following the Beer-Lambert law. The DNS assay effectively measures reducing sugars, and diluted samples show lower absorbance values, confirming the reliability of the method. Cooling the samples before measurement also improved the accuracy and consistency of the results. Overall, the experiment demonstrates that the DNS method is a reliable technique for quantitative analysis of reducing sugars.
From this practical section, we have learned how the spectrophotometry and the DNS determination method can be used to determine the reducing sugar concentration by measuring the absorbance at 575 nanometer. The practical also helped us to understand the Beer-Lambert law, where higher glucose concentration give the higher absorbance value. Besides that, we learned the reaction mechanism of DNS reagent with reducing sugar, which produce a reddish-brown color complex. Aerosol salt is introduced to stop the reaction and stabilize the color development. This practical also improves our laboratory skill, such as preparing several dilution, using micropipette accurately, and operating a spectrophotometer correctly. In addition, we learned that the factors like temperature, boiling time, cooling process, and pipetting accuracy can affect the accuracy of the experimental result.
One of the primary challenges encountered during this experiment was managing the extreme temperature sensitivity of the DNS reaction. Because the final absorbance is highly dependent on the temperature and the time of measurement, any minor differences in how long each trial was allowed to cool before being read at 575 nanometers likely caused the discrepancy we saw in our data. Additionally, we faced a technical challenge regarding the Beer-Lambert's law. Several of our sample readings exceeded the ideal absorbance range of 0.7. As noted in the principle of spectrophotometry, readings above these thresholds tends to underestimate the real absorbance, making it difficult to achieve perfect quantification without performing multiple time-consuming dilution. Finally, the physical act of micropipetting volume as low as 50 microliters requires significant precision. Even a small air bubble could lead to inaccurate sugar concentration. To improve the experiment for future sessions, we could adopt a greener route for sugar quantification. Modern research suggests using organic proton transfer salts like AED, which would allow us to eliminate toxic additives like phenol and Rochelle salt, making the lab safer and more environmentally friendly. We also suggest replacing the traditional boiling water bath with microwave heating. This adaptation can reduce the reaction to just 4 minutes and provides a more uniform distribution of thermal energy, which would help solve the trial-to-trial variance we experience. Furthermore, moving the assay to microtighter plates would transform it into a high-throughput technique, allowing more replicates in a shorter time frame while significantly reducing waste. Lastly, implementing a standardized pre-dilution protocol for commercial drinks would ensure all our absorbance values stay within the most accurate detection range.